This video is about the Corynebacterium diphtheriae. They are gram-positive rods. They are generally non-capsulated and they are non-motile. And the shape is club-shaped with the swellings at the ends. So characteristically, there are two features. They are Chinese letter shape like V and L shaped, and it is due to the cell division, bacterial cell division. The daughter cell will be at the acute angles or at the right angle. So it is called as snapping type of division, and it has metachromatic granules at the ends of, or poles of the bacilli. We can see like this. It is granular structure, and it generally stores the polymeta phosphates, and it is strongly gram positive, so it can be seen as darkly stained part, the poles. Next we have the virulence factors. Virulence factor is generally diphtheria toxin, the toxin that is produced by the bacteria. It generally has two fragments, A and B. So B fragment helps in binding to the cell, binding to the host cell receptors that are present on the cells, so which helps in entry of the fragment A. So B will bind and help the entry of fragment A. So this, after entry of fragment A of the toxin, A does the ADP ribosylation of the elongation factor 2. So we have something called elongation factor 2. So this is ADP ribosylated. So what happens is it will inhibit the elongation factor 2, and it inhibits the protein synthesis by inhibition of translation. So this elongation factor 2 is inhibited. It inhibits translation and it inhibits the protein synthesis. This is about the virulence factor. Next, coming to the pathogenesis and clinical manifestations. Due to diphtheria toxin, generally pathogenicity is seen, but it is not due to bacteremia. But bacteria can cause skin lesions, okay, there are punched out ulcerative lesions seen on the skin in some individuals with Corynebacterium diphtheria infection. And the bacilli are generally non-invasive, so they are present at the local site like pharynx, and they secrete the toxins which generally spread in the blood, and it can cause three different conditions. For they are respiratory diphtheria, cutaneous diphtheria, and systemic complications. So first, coming to the respiratory diphtheria. In respiratory diphtheria, there is Faucial diphtheria, which means that diphtheria toxin causes the inflammatory reaction. So this inflammatory reaction will cause the necrosis of the epithelium, and there is exudate formation. So what happens is, due to the necrosis of the epithelium, there is formation of mucosal ulcers. So these mucosal ulcers are lined by pseudomembrane coat which is formed. It contains fibrin and RBC and neutrophils. So this is seen as a pseudomembranous coat on the generally in the mucosa of the mouth. So it bleeds on the removal. So it is very rich in the blood supply. So it is attached to the mucosa. So whenever we try to peel the pseudomembrane, it generally bleeds, and when this pseudomembrane gets highly extended into the larynx, it may be obstructing the airway, and it can cause fatal airway obstruction. So, and also respiratory diphtheria can be seen as massive tonsillar swelling. Bull neck appearance is seen. It is characterized by foul breath and thick speech and stridor. And you can see, I will show you an image of respiratory pseudomembrane. This is the pseudomembrane formation inside the mouth, and this is the bull neck appearance due to tonsillar swelling. Next, we have cutaneous diphtheria, that is the punched out ulcers mainly in the extremities, and they can be formed by non-toxigenic strain, as they are caused by the bacilli, not the toxin. Or yeah, you can see in this punched out ulcerative lesions present on their extremities. And in the systemic complications, we see other conditions like neurological manifestations, cranial nerve involvement, peripheral neuropathy and ciliary paralysis, and also we can see in heart myocarditis. And next we have lab diagnosis. So in laboratory diagnosis, we generally collect the specimen, and we do direct smear or culture media, and biochemical reactions we'll see, and diphtheria toxin demonstration we'll see. So coming to specimen, we generally collect the throat specimen or throat swab, and also we can take a piece of the pseudomembrane or we can take skin specimens in ulcerated lesions. So in direct smear we can see by gram staining we can see club shaped bacilli, and in Albert staining we can see the rods are generally green color, and the metachromatic granules are generally seen as blue-black appearance. And in culture media we can see in enriched media, that is blood agar, small circular white colonies can be seen, whereas in Loeffler serum slope we can see glistening white with yellow tinge on the surface. And we can see in here, this is the Loeffler serum slope, so you can see glistening white type of colonies and tinge. Next we have selective media, that is potassium terolate agar, PTA, which is lysed horse blood is used. So what happens is the potassium terolite is converted into metallic terullium, which is black in color, by Corynebacterium diphtheria. You can see here black color colonies are formed of potassium terolite agar plate, which shows the black color colonies. These black colonies are due to formation of metallic terullium, which is converted by Corynebacterium diphtheria. Next, coming to biochemical reactions, we have catalase positive, urease negative, oxidase negative. Next, for the toxin demonstration, we do the Elix gel precipitation test. In this, we generally use 20% horse or sheep serum agar plate. So first we prepare the agar plate, and then we take out a small strip of filter paper which is dipped in the antitoxin and placed in the agar plate. And next, perpendicular to this antitoxin, the Corynebacterium diphtheria is stroked, and we can see after 1 to 2 days there is formation of like lines or precipitation bands. We can see due to diffusion of antitoxin towards the toxin released. Toxin will be transported in the agar like this, and antitoxin will be transported here, and there is formation of precipitation bands. These precipitation bands are formed, we can see only in toxigenic Corynebacterium diphtheria, whereas in non-toxigenic strains we can't see these precipitation bands. Okay, the toxin diffuse and meet the antitoxin, and precipitation lines are seen. This is about the Elix gel precipitation test. And coming to treatment, anti-diphtherial serum or ADS antitoxin is given, and antibiotics like penicillin, erythromycin can be given. And